Dear student, I am Dr. Prakash Mungli, Professor of Biochemistry. In this lecture, I will be explaining you genetic code, mutations and translation process. Now let's get into the details. The second stage in the gene expression is translating the nucleotide sequence of a messenger RNA molecule into an amino acid sequence of a protein. The genetic code is defined as the relationship between the sequence of nucleotides in DNA and the sequence of amino acids in a protein. And each amino acid is specified by one or more nucleotide triplets which are referred as codons in the DNA. And during translation, mRNA acts as a working copy of the gene in which codons for each amino acid in the protein have been transcribed from DNA to messenger RNA. So the tRNA serves as an adapter molecule that couples the codons in mRNA with the amino acids they each specify, thus they aligning them in an appropriate sequence before a peptide bond formation will go on. Now the translation, it takes place on ribosomes, complexes of a protein and rRNA that serves as the molecular machines coordinating interaction between mRNA, tRNA, the enzymes and the protein factors that are required for protein synthesis. And many proteins further will undergo post-translational modification as they prepare to assume their ultimate roles in the cell. And this is how the genetic code that is present in a DNA will be transcribed by an RNA polymerase to make a messenger RNA, and the messenger RNA will be decoded. The codons present in the messenger RNA will be decoded by a tRNA molecule, and thereby the specific amino acids are brought in as per the sequence that is present there, and that's how a specific protein is made to alter the physiology of the cell, and that's what is the translation process there. Now coming to the genetic code, each codon consists of 3 bases referred as triplets, as you see here in this particular table. So there are total 64 codons, you can count them in this particular table. So there are 64 codons there. So they are all written in 5' to 3' direction. When you read them from right to left, it is 5' to 3' direction. So out of 64 codons, 61 codons will code for amino acids. The other 3 codons, that is UAA, UGA and UAG. These are the stop codons or nonsense codons. These stop codons or nonsense codons, they terminate translation. And also there is one start codon which is also referred as initiation codon, and the initiation codon is AUG, which is coding for methionine. And protein synthesis begins with this methionine in eukaryotes, and in prokaryotes it begins with formyl methionine. And also note that the coding is unambiguous. That means each codon specifies no more than 1 amino acid, although one amino acid may have more than 1 codon. The code is degenerate, meaning more than 1 codon can specify a single amino acid. All amino acids except methionine and tryptophan have more than 1 codon. For those amino acids having more than 1 codon, the first 2 bases in the codon are usually the same. The base in the 3rd position is the one that varies. Also note that the code is universal. That means the codon is same in all organisms, with some minor exception that occurs with mitochondria. And also code is commaless, it is contiguous. There is no space or commas between the codons on mRNA. And neighboring codons on a message are non-overlapping. So these are all the characteristics of codons. Okay, so as you see here in the table, so Find out the stop codons, and that is UGA, UAA and UAG. These are the stop codons. And the start codon is AUG that I am marking here. Look for an amino acid that has got more than one codon. Look at alanine here, alanine, which has got 4 codons. Okay? And if you carefully observe that those 4 codons, the first 2 letters in the codon is same. As you see, GC, GC is all same in the 4 codons that are coding for alanine. The last letter is the one that differs in all of these 4 codons which are coding for alanine. There are 64 codons. 61 codons code for amino acids and 3 codons are nonsense codons, and they are UAA, UGA and UAG. AUG is the start codon for coding for methionine in eukaryotes, whereas formyl methionine in prokaryotes. Each codon code for a specific amino acid, meaning that code is unambiguous. Single amino acid may have more than one codon, that means code is degenerate. Other than methionine and tryptophan, all other codons have more than one codon. First 2 bases in the codon are same in the amino acid having more than one codon, and the third base is the one that is different, which I have explained with the alanine. Genetic code is universal. The genetic code is contiguous, that means there is no comma in the mRNA. Genetic code is non-overlapping. So these are the characteristics of codons. Now let me explain you mutations. A mutation is any permanent heritable change in the DNA base sequence of an organism. This altered DNA sequence can be reflected by changes in the base sequence of mRNA and sometimes by changes in the amino acid sequence of a protein. And mutations can cause genetic diseases. They can also cause changes in enzymatic activity, nutritional requirements, antibiotic susceptibility, morphology, antigenicity, and many other properties of a cell. A very common type of mutation is a single base alteration, also referred as point mutation. A transition is a point mutation that replaces a purine-pyrimidine base pair with a different purine-pyrimidine base pair. For example, an adenine-thymine base pair becomes a guanine-cytosine base pair. A transversion is a point mutation that replaces a purine-pyrimidine base pair with a pyrimidine-purine base pair. For example, adenine-thymine base pair becomes thymine-adenine or cytosine-guanine base pair. So here a purine is replaced by pyrimidine or pyrimidine is replaced by a purine. Now let me explain you common types of mutations that are seen in a DNA. So the figure here, it is showing different types of mutations that are commonly seen in DNA. So that is silent mutation, missense mutation, nonsense mutation and frame shift mutation. So the mutations are often classified according to the effect they have on the structure of genes protein product. The change in the protein structure can be predicted using the genetic code table in conjunction with the base sequence of DNA or mRNA. So the table here which shows a silent mutation, it is a new codon that specifies the same amino acids. So the effect on protein is none, because that's why it is called as a silent mutation. Whereas a missense mutation is a new codon specifies a different amino acid. That means a possible decrease in a function of a protein may be seen, or there may be a variable effect in that function of a protein. Nonsense mutation is a new codon is a stop codon. That means a shorter than normal protein is being synthesized, and usually it is non-functional. And the frame shift mutation is an addition or deletion of a base in the mRNA sequence of codons there. Usually, this protein will be non-functional and often it is shorter than normal. And then the large segment deletion, which is because of unequal crossover in meiosis, it leads to loss of function of a protein molecule. And then we have like 5' splice site or the 3' splice site mutation is it can lead to variable effects ranging from addition or deletion of few amino acids to deletion of an entire exon is seen leading to altered protein function and no function of a protein. And then we have trinucleotide repeat expansions. So here expansion in coding region causes protein product to be longer than normal and it is usually unstable. And disease of then shows an anticipation in pedigree. That's what is a trinucleotide expansion there. Coming to large segment deletions. A large segment of DNA can be deleted from a chromosome during an unequal crossover in meiosis. A crossover or a recombination between homologous chromosomes is a normal part of meiosis, number 1, that generates a genetic diversity in reproductive cells like eggs and sperm. Largely, it is beneficial. In a normal crossover event, the homologous maternal and paternal chromosomes, they exchange equivalent segments, and although the resultant chromosomes are mosaics of maternal and paternal alleles, no genetic information has been lost from either one. On rare occasions, a crossover can be unequal and one of the two homologous loses some of its genetic information. Example, in alpha thalassemia. Alpha thalassemia is a well-known example here for a genetic disease in which an unequal crossover has deleted one or more alpha globin genes from chromosome number 16. So, Cri-du-Chat syndrome, where mental retardation, microcephaly, wide set eyes and characteristic kitten-like cry, it results from a terminal deletion of a short arm of chromosome number 5. This is about a large segment deletion. Coming to mutations in splice sites. Mutations in splice sites affect the accuracy of intron removal from hnRNA during post-translational processing. If a splice site is lost through mutation, spliceosomes may delete nucleotides from the adjacent exon or leave nucleotides of the intron in the processed mRNA, or use the next normal upstream or downstream splice site, deleting an exon from the processed mRNA. Mutation in splice sites have now been documented in many different diseases. That includes beta thalassemia, Ghostzer's disease, Tay-Sachs disease and all that. That's about mutation in splice sites. Coming to trinucleotide repeat expansion. So the mutant allele in certain diseases like Huntington disease or Fragile X syndrome, as it is given here in the table down here, or myotonic dystrophy, they differ from their normal counterparts only in the number of tandem copies of a trinucleotide. In these diseases, a number of repeats often increases with successive generations and correlates with increasing severity and decreasing age of onset of a disease. And this phenomenon in trinucleotide repeat expansion disorders is referred as anticipation. For example, in the normal Huntington allele, there are 5 tandem repeats of CAG in the coding region, and affected family members may have 30 to 60 of these CAG repeats. And the normal protein contains 5 adjacent glutamine residues coming from CAG, whereas the proteins encoded by the disease-associated alleles have 30 or more adjacent glutamines. And the long glutamine tract makes the protein abnormal and extremely unstable. A major clinical manifestation of trinucleotide repeat expansion disorders is neurodegeneration of specific neurons. And the expansion of trinucleotide repeat in the mutant allele can be in a coding region or in an untranslated region of the gene. So, some of the trinucleotide repeat disorders given here, we have like Huntington disease, spinobulbar muscular dystrophies, and that ones that are going on in untranslated region, Fragile X syndrome, myotonic dystrophies, and Friedreich's ataxia. Coming to amino acid activation and codon translation by tRNAs. tRNAs are the adaptor molecules. So tRNAs, they decode mRNA codons during the translation process. And once amino acid is activated there, so tRNA binds with a specific amino acid at the 3' end with energy-rich bond, as it is shown here in the figure. And tRNA has an anti-codon sequence which is complementary and anti-parallel to the codon on mRNA, and that's how it is going to read that codon on mRNA, and accordingly is going to arrange a specific amino acid as per the sequence, and later the peptide bond formation will go on. Now let's look at a translation process or a protein synthesis. Protein synthesis occurs by a peptide bond formation between successive amino acids whose order is specified by a gene, and thus that is by an mRNA. The formation of a peptide bond between a carboxyl group of one amino acid and the amino group of another amino acid is shown here in this particular figure down here. An example that is taken here is methionine, which is carboxyl group of methionine is going to interact with the amino group of tyrosine and making a peptide bond. Now, during translation, the amino acids are attached to the 3' end of the respective tRNAs, and the amino acid tRNAs are situated in the P and A sites of the ribosomes. So, that's what is shown here in the figure. So, there is a amino acid tRNA in the P site, which is the first one, and then the amino acid tRNA is coming into the A site, which is bringing a new amino acid, and then the carboxyl group of the first amino acid, which is part of the tRNA, is going to make a peptide bond with the amino group of the incoming amino acid that is there in the A site. So, the P site amino acid carboxyl group and A site amino acid amino group that participate in the peptide bond formation. So, notice that the peptide bond forms between the carboxyl group of the amino acid or the growing peptide in the P site and the amino group of the next amino acid in the A site, which is incoming. And proteins are synthesized from the amino to carboxy terminus, because in the methionine here, that's the amino group on the left side, and all other amino acids are which are coming and making the peptide bond with their amino groups and their carboxyl group on the right side. So, as this process goes on, so there will be amino group on the left side and carboxyl group will be there on the right side. Now, let me explain you steps of translation. Translation occurs in the cytoplasm of both prokaryotes and eukaryotic cells. In prokaryotes, ribosomes can begin translating the mRNA even before RNA polymerase completes the transcription process. That means translation in prokaryotes is a co-transcriptional translation. So, in eukaryotes, a translation and transcription are completely separated in time and space, with transcription going on in the nucleus and translation is going on in the cytoplasm. The process of protein synthesis, it occurs in 3 stages. That is, it involves initiation, elongation and termination. And special protein factors for initiation, elongation and termination, which are referred as the releasing factors, as well as GTP are required for each stages. Now, let's look into the details on initiation stage of protein synthesis. As it is shown in the figure down here, the small ribosomal subunit binds to the mRNA. In prokaryotes, the 16S ribosomal RNA of the small subunit binds to this Shine-Dalgarno sequence in the 5' untranslated region of mRNA. Whereas in the eukaryotes, the small subunit binds to the 5' cap structure and slides down the message to the first codon, that is AUG. The charged T initiator tRNA becomes bound to the AUG start codon on the message through the base pairing with its anticodon. The initiator tRNA in prokaryotes carries F-methionine, that is formyl methionine, whereas the initiator tRNA in eukaryotes, it carries only the methionine. The large subunit binds to the small subunit, forming a completed initiation complex now. And there are two important binding sites on the ribosome called P site and A site. P for peptidyl site is the site on the ribosome where F-methionine tRNA initiator tRNA initially binds there. After formation of the first peptide bond, the P site is a binding site for the growing peptide chain. The amino cell site, that is the A site, binds each new incoming tRNA molecule carrying an activated amino acid. So this is how, the P site, original amino acid, initiator amino acid like AUG coding for methionine, it will be there. And A site is the new amino acid that is coming in as per the codon present in mRNA. Peptide bond formation goes on and will move to the P site, and the A site is empty and new amino acid is brought in by tRNA and make a peptide bond. It will move to the P site, A site is empty. Like this, it goes on and on. And that's how initiation will be going on. Now let's move on to see elongation. Elongation is a 3-step cycle that is repeated for each amino acid added to the protein after the initiator methionine. Each cycle uses 4 high energy bonds, 2 from ATP used in the amino acid activation to charge tRNA and 2 from GTP. So during elongation, the ribosome moves in the 5' to 3' direction along the mRNA, synthesizing protein from amino terminus to carboxy terminus. The 3 steps are: a charged tRNA binds to the A site. A particular amino acyl tRNA is determined by the mRNA codon aligned with the A site there. And then the peptidyl transferase, an enzyme that is part of a large subunit of ribosome, forms the peptide bond between the new amino acid and the carboxyl end of the growing peptide chain, which is there in the P site. The bond linking the growing peptide to the tRNA in the P site is broken, and the growing peptide attaches to the tRNA located in the A site. Okay. Now, in the translocation step, the ribosomes moves exactly 3 nucleotides, that is 1 codon, long message. This moves the growing peptidyl tRNA into the P site and aligns the next codon to the translated with the empty A site. So that's how the movement will go on, and A site will be empty for incoming tRNA to bind with the activated amino acid. So in eukaryotic cells, elongation factor 2 is used in the translocation, and that can be inhibited or inactivated through ADP ribosylation, and that's how Pseudomonas and diphtheria toxins can inhibit protein synthesis. 𝐒𝐫 and 𝐒𝐫-like toxin clip and adenine residues from 28S ribosomal RNA in the 60S subunit, stopping the protein synthesis in eukaryotic cells. So this is how elongation will be going on, and some of the toxins which can act on the elongation process. Coming to termination process. Now let me explain you termination process. When any of the 3 stop codons moves into the A site, the peptidyl transferase, with the help of releasing factor, hydrolyzes the completed protein from the final tRNA in the P site. So once this happens, mRNA, ribosomes, tRNA and all the factors will be dissociated and released, and they can all be reused for additional protein synthesis. And this is what happens in the termination, and the new polypeptide chain is released. So there is something called as polysome. A messenger RNA molecules are very long compared with the size of a ribosome, allowing room for several ribosomes to translate a message at the same time. So because ribosomes translate mRNA in 5' to 3' direction, the ribosome closest to the 3' end as the longest nascent peptide, and the polysomes are found free in the cytoplasm or attached to the rough endoplasmic reticulum depending on the protein being translated, and that's what is about a polysome there. Now let me explain you a little bit about inhibitors of protein synthesis. Inhibitors of eukaryotic translation include cyclohexasamide, diphtheria, and Pseudomonas toxins. Certain antibiotics like chloramphenicol also inhibit mitochondrial protein synthesis but not cytoplasmic protein synthesis because mitochondrial ribosomes are similar to prokaryotic ribosomes. Now let me explain you a little bit on site of translation. Translation can occur in free ribosomes or on the rough endoplasmic reticulum. Although all translation of eukaryotic nuclear genes begins on ribosomes free in the cytoplasm, the protein being translated may belong to other location. For example, certain proteins are translated on ribosomes associated with the rough endoplasmic reticulum, and they include secreted proteins, proteins inserted into the cell membranes, or proteins going into the lysosomes as the lysosomal enzymes. Our proteins translated on free cytoplasmic ribosomes include like cytoplasmic proteins or mitochondrial proteins which are encoded by nuclear genes. So this is about the genetic code, mutations and translation process. Thank you.